NextGen Science Key.
Hello, so today I'm going to discuss about digestion and absorption. That is the structure of the digestive system, the oral cavity, the tongue, the oropharynx, the esophagus, the food pipe, stomach, the duodenum, which is the pancreas attached to duodenum, the jejunum, which is the lower part of the small intestine, colon, then the anal canal, which is the anus. The digestive system consists of two things. One is long hauler muscular tube, which is also called canal or tract, called the gastrointestinal tract, about 5 meters long. Accessory glands are other glands, which is salivary gland, liver, gallbladder, and pancreas. The GIT consists of oral cavity, the pharynx, the esophagus, the stomach, the small intestine here from the duodenum, jejunum, the small intestine, the large intestine, the rectum, and anus. So that is the structure of the GIT, how it looks. It has the villi. The villi have the blood capillaries supplied to them here, which assist in absorption, and there are so many to increase the surface area, lymphatic vessels, which fight the microorganism. The main functions of the digestive tract is for motility, that is to propel ingested food from mouth towards rectum, secretion of juices, which consist saliva. They assist in digestion and absorption. The third thing is digestion, that is the breakdown of food. The fourth thing is absorption. Nutrients and electrolytes, water are absorbed or transported from lumen or the GIT to the bloodstream. This is food. The food is broken down by digestion. Then absorption is from the digestive tract to the blood. From the digestive tract to the blood through the intestinal fluid absorption. Then secretion is movement from here, the digestion, or from here to the blood. That is secretion. Then motility, movement. This food moved from here, going to the other part. So motility of the mouth, that is how the food moves to the mouth. There are two things that takes place there. The first one is chewing. The food is broken down by chewing, by the help of teeth. That breaks the food into smaller particles to be easily swallowed to increase surface area for absorption. It's also exposed food to salivary amylase enzyme. In the mouth, you have the salivary amylase enzyme, which begins the digestion of starch. It helps digestion of all types of food, especially cellulose containing food, which are vegetables. The second thing is swallowing. Swallowing is transport of food from mouth to stomach. And there are three steps. And one is buccal phase. Food is pushed back into the pharynx of the mouth. Food is pushed back into this part called the pharynx of the mouth. That is the first step. And the second step, the pharyngeal phase, is where the food is put through the pharynx to esophagus. And we have the epiglottis here, which prevents the food from getting into the trachea. The third phase is esophageal phase. Here the food passes through the esophagus, which is also called the food pipe, to the stomach through the process called peristalsis. The esophagus is 25 centimeter tube. It is guarded by two sphincter muscles. And these two sphincter muscles are esophageal sphincter, contract here, sphincter muscles, and you have the contracted muscles here. So we have the sphincter, relaxed muscle. So we have esophageal sphincter and sphincter relaxed muscles. Upper esophageal sphincter prevents air from getting into the GIT. So the upper part prevents air from getting into the GIT. The lower esophageal sphincter prevents gastric contents from entering the esophagus from the stomach. This one prevents the food from getting back to the esophagus. So the esophageal peristalsis sweeps down the esophagus. The esophageal peristalsis, that is the movement of food from here to here. It sweeps down the esophagus. So the stomach consists of fundus, body, and pylorus. Fundus, here it is the upper part. Then we have the body here. Then you have the pylorus. Here, the proximal area, fundus and the body have thin wall and contract weakly and frequently. Holds large volumes of food to store food because of the receptive relaxation here. They have thin wall and they hold food. The diastole area has a thick wall and with strong and frequent peristaltic contraction that mix and propel food into the duodenum. This duodenum is the upper part of the smaller intestine. Also, diastole area is responsible for gastric emptying into the duodenum. The diastole area is responsible for mixing of food and also the emptying of food into the duodenum. Two basic motility patterns exist: segmentation and peristalsis. The significance of segmentation and peristalsis motility of small intestine serves three functions: mixing contents with enzymes and other secretions, which helps in digestion; maximizing exposure of contents to membranes of the intestinal cells, which helps absorption and digestion; propulsion of contents into large intestine. So that is the significance of segmentation and peristalsis. Segmentation movement, segmental contraction are responsible for mixing and cutting. Contraction takes here, yes, and there's relaxation here. So as it contracts, it cuts the food into smaller boluses. That one is segmentation, and in segmentation, there is no movement because the food is just broken down to bolus parts. Peristalsis, peristaltic contraction are responsible for forward movement. As you can see here, it contracts, then here it is the bolus, and it passes. Here it contracts, then the food moves, then it contracts here, the food moves. Bolus will tend to move to the right, to this direction. Peristalsis is for bolus movement, but the segmentation is there for the breakdown of food. Segmentation in large intestine causes the contents to be continuously mixed. Mass movement propels the contents of one segment of large intestine into the next downstream movement. Bifurcation involves involuntary reflexes and voluntary reflexes, evacuation of colonic content through the anal canal. Secretory function, secretions of GIT. The total volume of the GIT secretion is about 6 to 8 liters a day. Secretion arises from specialized cells lining to the GI tract, which is the gastrointestinal tract, the pancreas, the liver, and the gallbladder. GI secretions functions to lubricate. That is the first function of the GI secretions: lubricate water and mucus. Sterile here, which sterilize, which is the HCl. Then neutralize hydrogen carbonate and digest. Those are the enzymes. So salivary glands, there are three pairs of salivary glands. One is the parotid. Parotid is here, just closer to the ear. Then we have sublingual. As the term lingual, it means the tongue. So this one is found under the tongue, sublingual gland here. Submandibular. Submandibular is just found below the jaw. So you can see functions of saliva is to lubricate, cleanse the oral cavity, and dissolves chemicals, suppress bacterial growth, that is for antiseptic. So the first one is lubricates. Saliva is made up of water and mucus. It cleans oral cavity, dissolves chemicals, suppresses bacterial growth, digests starch by amylase, salivary amylase, which assists in digestion of starch. Cerebral cortex, that is found in the brain. So this one is the regulation of the salivary. So salivary center is the medulla, which controls the secretion of saliva. So pressure receptors and chemoreceptors in the mouth, they trigger the salivary. Then there is unconditioned reflex, which include the autonomic nerves. When a person smells something good, something delicious, it will trigger the medulla, which the medulla will make the salivary gland to produce more salivary to cause the salivary excretion. So as you can see, GIT excretions in the stomach. The stomach, this is the structure of the wall of the stomach, the lumen, the mucous neck cell here, the parietal cell here, the enterochromaffin-like cell here, the chief cells here, D cells, the D cells, these ones and this one, and this one, the D cells, then the G cells. So the G cells are called the gastrin, which stimulates gastric acid secretion. D cells, somatostatin, inhibits gastric acid secretion. So this one is the antagonist of this one. So the D cells inhibit the gastric secretion, while the G cells stimulates the gastric acid secretion. Then the chief cells have the pepsin and gastric lipase. So digests protein. The pepsin digests proteins. So digestion of proteins takes place in the stomach. Gastric lipase digests fats. Enterochromaffin-like cells have histamine. Histamine causes allergy, stimulates gastric acid secretion. So this histamine, anti-ulcer drugs, they are drugs which has two receptors. which antagonize this one, production of this histamine. So stimulates gastric acid secretion. The parietal cell, which is the gastric acid, HCl. So this HCl as for antiseptic. So activates pepsin, kills bacteria. Then intrinsic factor complexes with vitamin B12 to permit absorption. The mucous neck cell, which produces mucus, physical barrier between lumen and epithelium. Bicarbonate buffers gastric acid to prevent damage of epithelium. So the main ones are those ones, mucus cells, then parietal cells and chief cells. Chief cells, pepsinogen, which digest proteins, and G cells are the antagonists of D cells. So functions of the gastric HCl activates pepsinogen into pepsin. And pepsinogen is in active form to prevent that pepsin from digesting the walls of the stomach. Number 2, provides optimum pH for action of pepsins. So pepsins, they work well while in acidic pH. Denatures protein denaturization, helping digestion. Proteins are broken down by denaturization. Then it kills bacteria in the food, help iron 2 and calcium absorption, promotes pancreas, small intestine, and bile secretion. HCl, this one is an acid. Then here we have the pepsin. The pepsin is in cloudy form. So HCl, when added to pepsin, it denatures the structures of pepsin, making it more clearer than the first one. So as you can see here, it has assisted in digestion of pepsin. So pepsinogen, HCl activates and it denatures the pepsinogen to pepsin, which breaks the protein to peptone, which is more simple for digestion and absorption. Soluble and insoluble mucus are secreted by cells in the stomach. Soluble mucus mixes with the contents of the stomach and helps to lubricate chyme. Insoluble mucus forms a protective barrier against the high acidic of the stomach content. Intrinsic factor help absorption of vitamin B12. That one is for HCl. Regulation of gastric secretion. So vagal scent of medulla here, when the food is taken inside, it triggers the stomach to produce the HCl here. So gastrohistamine stimulation here, the vagal reflexes and local nervous secretory, the parasympathetic excite pepsin and acid production. So pancreas, that is the second accessory gland here. So the pancreatic isolates of Langerhans here, cells secrete hormones. There's pancreatic duct here. Then there's lobules. As you can see, pancreas is leaf-shaped. So pancreas has two functions, endocrine functions, but it secretes insulin and glucagon from the islet of Langerhans. This insulin and glucagon, they are used in regulation of blood sugar levels. Then exocrine secretion of pancreatic juice. It has two components, aqueous and enzymatic components. Aqueous component contains hydrogen carbonate. It's important for neutralizing stomach acid, which is HCl in the duodenum, so pancreas enzymes can function properly. Enzymatic component is essential for proper digestion and absorption of carbohydrates, fats, and proteins. Pancreatic enzymes include trypsin, chymotrypsin, lipase, and amylase. So pancreatic amylase starch broken down by pancreatic amylase to maltose and maltose. Then fat triglyceride is lipase, and colipase is broken down to monoglyceride and fatty acids. Trypsinogen, which is enterokinase enzyme, breaks to trypsin protein. This trypsin activates proteins to polypeptide. So chymotrypsinogen to chymotrypsin. So carboxybutase to the polypeptidase to amino acid. Enterokinase here, there is chymotrypsinogen and chymotrypsin. So chymotrypsinogen is broken down to, is activated to chymotrypsin. So that is chymotrypsin, and trypsinogen is activated to trypsin. So this trypsin is the active part. So regulation of pancreatic secretion, the brain, which has the vagus nerves, by the presence of food in the stomach, it activates the secretion of that pancreas. So presence of acid in the duodenum causes release of secretin. Presence of fats in the duodenum causes release of cholecystectin. So this Secretin is for the breakdown, is for the production of hydrogen carbonate. Then presence of fats, this cholecystokinin is for the breakdown of fatty acids. Vagal stimulation causes release of pancreas enzymes, sphincter or OD. This one is where it is connected. And then secretion causes release of bicarbonate secretion. CCK causes secretion of enzymes. So that is the control of that one. So secreted when the pH here lower goes down, or when the pH is lower, so it produces secretin, which produces the hydrogen carbonate and regulate the pH of the duodenum. Then cholecystokinin, when there are more fat, cholecystokinin is produced to regulate, to break down the fat. Then that way, so liver and gallbladder. As you can see, this is the liver and the gallbladder. So the liver is for the production of bile. Then the liver is for the storage of bile. Liver functions the liver metabolic regulation, to absorb nutrients and vitamins, release nutrients as needed. Hematological regulation. So 1 is metabolic regulation, 2 is hematological regulation, and 3 is production of bile. So to absorb nutrients, that is for metabolics, to absorb nutrients, vitamins, release nutrients as needed. Hematological regulation, this is for plasma protein production, then remove all red blood cells, then this production of bile. This required for fat digestion and absorption. Secretion from duodenal gland and intestinal gland. Secretory volume is 1 to 3 liters per day. It contains inorganic ion, mucoprotein IgA, here IgA, various enzymes, and e.g. the enterokinase. Function, protective effects. This now, that one was for the liver here. Then you have small intestine secretion, which consists of secretion of duodenal gland and intestinal gland. Then you have the secretory volume of 1 to 3 liters a day. It contains inorganic ion, mucoproteins, IgA, various enzymes, e.g. enterokinase. Then the functions is protect effect by mucus, digestion of enzymes such as peptidase, sucrase, lipase, and dilution. As you can see here, dipeptides are broken down by dismutase to amino acids. Then maltose is broken down to maltase, glucose plus glucose. This maltose is combination of glucose and glucose. So lactose, it is broken down by enzyme lactase to glucose and galactose. And then sucrose is broken down by enzyme sucrase to glucose and fructose. Then nucleotides is broken down by enzyme nucleotides to nucleosides. Then nucleosidase breaks down sugars to bases. Then di and monohydrates are broken down by enzyme lipase to fatty acids and glycerol. Then secretion in the large intestine, there's colonial alkaline secretion to neutralize acids produced by the intestinal bacteria. Excretion of mucous proteins for protection, lubrication of fecal matter. Vitamin B and K absorption made by bacterial flora and colon. So that was digestion. Digestion and absorption. Digestion is the process essential for conversion of food into smaller simple forms, which are the mechanical digestion here.